Aya Bassiouni, German University in Cairo, Egypt.
Okay. Imagine every day you enter a battlefield, ready to fight and willing to beat your opponent, yet you end up paralyzed and powerless. Unfortunately, this has become the reality for patients suffering with hepatocellular carcinoma. Hepatocellular carcinoma is the end stage of liver disease, rendering it the second leading cause of cancer-related mortality. The outspread of this disease has been vastly escalating globally. However, the outcome of therapeutic modalities is still far from satisfactory. This is owed to the disease's high recurrence rate, exhaust of chemotherapy, and rejection of organ transplantation. And thus, it was necessary to look for a new therapeutic approach. So what if the solution is much closer than where we are looking? What if the answer lies where the disease is, inside our own bodies, where we have the immune system, the weapon, the striking warrior that is eager to combat this disease? This battle of the immune system is actually quite interesting, because in normal healthy individuals, the NK cells is shed for the green light activation upon recognition of the tumor cells. And thus, they release cytotoxic effector molecules and eventually cause eradication of the tumor cells. However, in hepatocellular carcinoma, these immune cells are defective, shedding the red light for NK cell inhibition, turning the balance in favor of the tumor cells, and thus they win the battle. So from here, the idea of immunotherapy burst, where my aim was to correct these immune cells by fine-tuning molecules known as microRNAs. These are small endogenous molecules and has been recently approved as drugs. My idea mainly focused on a novel promising microRNA known as LAT7A. So first, I screened the levels of this microRNA in the NK cell, in the tumor cells, where it was found to be downregulated. Upon forcing the expression of this microRNA, it caused shutting down of several signaling proteins and thus eradication of the tumor cell. However, in NK cell, screening the levels of this microRNA was found to be upregulated. Yet forcing the expression of this microRNA caused decreasing the release of the cytotoxic effector molecules release and decreasing the capability of the NK cell to combat the tumor cell. My aim alongside the research group is to apply this research in clinic through increasing the levels of this microRNA in tumor cells and thus halting tumor progression, and decreasing its level in the NK cells, and thus giving it the capability to be able to combat the disease. And for that, we aim to break the wall of hepatocellular carcinoma. Thank you.